probably see more than we want to. Well, you're an outstanding scientist, nationally recognized, right, and female, right, and so there we go, right. And so, but yeah, no, fair enough. You got to be able to say no. You got to say no. How's that social data community going? Data science. I had a really good chat with from Greece, the economist Kostas. Yeah, we had a Senate lunch, yeah, and there's some really great links there actually. Yeah. What about the environmental and biodiversity data side? Did that come up at all in this discussion? In that sense, yeah, we got to bring that together with our data science. Because you haven't heard from Dan Spielman either, because they're cooking up their own thing, but the environment and biodiversity is not part of it, right? So we got to make sure it's actually, because there it is. So will we have a chance to chat a little bit afterwards?
Right after the doctor's
Okay. Okay.
and then I can teach them.
All right.
So we'll have a, but I'm really happy yeah, let me introduce you and we'll get going.
You heard from Hugh back from Hugh at all?
No.
Okay.
No. Although I'm having a call with Rob McDonald on Friday. To talk about making it really safe for those people.
Okay.
About trying to do which people from those students specifically they're doing a bunch of stuff. A lot of it relies on people. Anyhow. Okay, well, good afternoon. We're going to get started. It's really nice today to introduce one of our affiliated faculty. Is this on? Can folks hear me in the back? Okay. Well, I was saying it's really nice to, thank you, Will. It's really nice to have one of our affiliated faculty give the Wednesday seminar today. Walter Jetz is a professor of ecology and evolutionary biology at Yale. He holds a DPhil from the University of Oxford, and for those of you who aren't familiar with the DPhil, it's Oxford's basic way of saying you have a PhD, but we have a special name for it if you get it from Oxford. His group works at the intersection of ecology, evolution, geography, and environmental sciences. Waltz's group uses big data-driven research, and they address the mechanisms underpinning large-scale variation of biodiversity and the resulting implications for conservation. His group uses a variety of different types of data, including remote sensing, phylogenetic, functional, and spatial temporal biodiversity data, combined with new modeling approaches and informatic tools. If you look on Walter's website, you can see a really large list of research activities, but almost an equally large list of outreach activities. I'm just going to list a couple of those. He's currently the director of Yale's Center for Biodiversity and Global Change and the lead of the Map of Life project, which is based here at Yale. Internationally, he holds a number of leadership positions as well. He's the co-lead of the GeoBond. So this is the Global Earth Observation Group, which is an international a network working group, and he specifically co-leads the group on BOND, which is the Biodiversity Observation Network within the Global Earth Observation Network. Walter is also chair of the IB Best Task Group on Indicators. So please join me in welcoming Walter Jetz.
Thanks, Karen. And it's a real pleasure to be here. I look at this building on a daily basis. We're over there in that kind of not so pretty gray building. It's got character, though. It's real wonderful to be an adjunct in FES, and it's one of the key reasons why I actually came to Yale, why I'm still at Yale, because I feel there is just an outstanding community of people here, and the work that I'm talking about today is work that's very much at the interface, I feel, of more basic ecology and evolutionary biology, and then the more applied aspects that are very much the theme here. And over the years, it's been wonderful getting to know a lot of the students here, and also the faculty. So really delightful to be here. And the topic today will be a map of life, but which Karen already mentioned. But I'll cast a much broader net to talk about biodiversity conservation in a policy arena more broadly. And this is obviously no news to you. This sort of very general introductory slide, the sort of thing we're kicking around a lot in IPBES, Intergovernmental Platform for Biodiversity and Ecosystem Services discussions, or Future Earth discussions, another one of these international groupings. Just the appreciation that if you really want to address ecosystem services and their relevance for human well-being, you got to start ultimately with the biodiversity, as that is what delivers many of the key ecosystem functions, and it's the drivers affecting biodiversity that we, as biologists in particular, as ecologists, need to think about as we want to do our piece in ensuring a more sustainable planet. Now, in the larger policy arena, international policy arena, we have these targets, and many of you will have come across obviously the Convention on Biological Diversity and their Aichi 2020 targets. It's actually the Conference of the Parties coming up in just a couple of weeks, and it will be interesting to attend that and contribute to the discussions there that will be very much about how we're going to move on from this and how we're going to possibly do better than this. Target 11, for example, the idea that we'd want to conserve a minimum amount of area for biodiversity to ensure the survival of species and their functions, and ensure a healthy planet by 2020. And so this is up for renegotiation, and in 2 years, so the next Conference of the Parties in Beijing, there will be finalization of some of the new targets. So this is an interesting time to think about how can we as scientists bring more pieces to the table, because up to this point, it's been very much an arena driven by strong lobbyism, by an engagement by several international NGOs, but we're in a new era now, in an era of big data, where we can put better data, better information, better science into this picture, and hopefully inform some of these targets, or at least how the processes around them, as they will be monitored and updated in the future. Now, the Sustainable Development Goals, also familiar to many of you, I'm sure. Here it's the goals 14 and 15 that are particularly relevant, and right now they're just adopting what the CBD kind of says about protected areas or conservation areas in general. And I'm sure that will be the case going forward as the CBD targets are getting renegotiated. So how we doing in all this? Well, you could argue that there has been substantial progress in around Target 11, one of these 20 targets, and especially recently in marine, terrestrial has been inching forward, and we are approaching the sort of percentages that are being mentioned there in the target. And this is out of work associated with the IPBES task group here, where we began to obviously compare how this plays out in different parts of the world. And unsurprisingly, it plays out in different ways, right? So in Africa or Central Asia, so this is all the way over to Siberia, even the amount of area projected on land is much lower than in other parts of the world. And then if you drill into one of these regions,
we find that countries differ dramatically, with Bhutan obviously, being one of these really leading examples. On the top there, South Asia, Bhutan, and many others doing underperforming against this target. And you may have noticed this piece about the representative of biodiversity, right? So obviously, as a biodiversity scientist, we should be lamenting up front the idea that you could just use area targets to represent biodiversity. It's in the written into the target that these areas are supposed to be representative, but then you look at places like Myanmar or Vietnam way down here, and we all know there's a hell of a lot of biodiversity in Myanmar and Vietnam, and certainly more than in the United Kingdom or other parts of Europe for that matter. So can we do, obviously we're not progressing in the way we ideally should, even with the target formulated as is, and going forward, could we perhaps set benchmarks that are more biologically sensible, given that there is now better information, better data that we scientists can bring to the table. So how are we doing in terms of our protected area network globally, or conservation areas more general? We shouldn't think of just protected areas, could be other types of management areas. How are we doing, and how can we do better going forward? And let me just spend a few minutes, a side excursion to this other large piece of research in our group that is more about the fundamental underpinnings of the spatial variation in biodiversity. That is an equally exciting arena with all sorts of new methods and technologies and data coming about, and where we are now able to come back to some of these really classic questions in biology, in biogeography. Here, Humboldt already musing ages ago that there is this latitudinal gradient in species richness, and not just richness, but also structure and grace of form, right? So we're talking a little bit about functional diversity, some of these other aspects of biodiversity beyond species richness. And this question is still one of the holy grails of ecology or biology in general. And here just a little personal story as to how I got into that research and into this field of work from starting out initially very much at the local scale. And for example, running around as an undergrad in West Africa, trying to understand how it is that you find so many more forest bird species there, or bird species in general, in this little patch in Ivory Coast, compared to, say, the places I was familiar with in Europe. So in very much kind of MacArthur type fashion, MacArthur, student of Hutchinson's, who used to be in the building just across there, one of the leading ecologists, and so was MacArthur, and he very much came in with that fundamental work about how by looking very closely at a community and looking at how species use different parts of a habitat differently, you can actually come to an understanding of the mechanisms supporting this coexistence. But as I ran around there and measured all sorts of things, I increasingly got to appreciate, well, just looking around here is not really going to explain that, is not going to answer that question for me. I'll have to scale up. I have to think more broadly. How is it that there are this many bird species in this larger region of West Africa? How is it that we have certain types of bird groups having diversified in Central Africa and the forest regions there, and then moved over into the western parts and overcome dispersal barriers? How is it that there are only under 2,000 bird species, well under 2,000 bird species in Africa, whereas there are many more bird species in South America? So as you compare a location in Africa with one in South America, that's one of the key things you need to appreciate. So I found myself more and more digging into some of the more historical and even evolutionary underpinnings of the biodiversity we see there today, and come to an appreciation of this scale dependence of the different processes affecting biodiversity at different scales, so historical and large scale all the way down to fine scale. And one thing that's really been characterizing research in my group is this attempt of trying to synthesize, trying to understand pieces of each of these levels so that we can really get to a better understanding of, well, the large scale, the broad scale, the macroecological patterns, but at the same time also the the fine-scale patterns and integrate among these processes. So system that we worked with a lot and are particularly excited about again is that of terrestrial vertebrates. So about 30,000 species in total. This is just their general patterns of species richness. We have a pretty good grip on their at least coarse grain. These are quite pixelated patterns of diversity. And what we're really excited about right now is the fact that we've been able to build phylogenetic trees or phylogenetic frameworks addressing each one of these groups, and that is something extremely powerful to now understand how the patterns of diversity in space that we see today have come about over these larger timescales. So this is thanks to work with collaborators and NSF funding and then students and postdocs in the lab group. So enough with that side excursion, if you will, about into the more basic research in our lab, but I thought I should give you a flavor of how this is really this key other piece, the underpinning of biodiversity patterns today and the methodology and the data that we require for that is something really exciting intellectually, scholarly, but it also sets us up in a really great way to now think about global biodiversity, yes, at the global scale, but also at really fine spatial detail from a conservation perspective. And that's kind of the more sort of thematic synthesis, if you will, that's happening in our lab group. And let's go really local and go to Ecuador here, where with Simon Queenborough a couple of years ago, we had the privilege to visit the Podocarpus National Park. So Simon did an amazing job leading the logistics here and organizing this all, and it was a real fun trip. I see one member at least who was along for that, so this was a previous class. And this was joint between FES and EB. It was sort of an experiment to see how we could all go to the field together and combine undergrads and graduates in a course that was really outstanding, and was partly outstanding because we got to meet with people like Renzo Palladines, who's running a local regional NGO there, Nature and Culture, that's all about figuring out how to get more land conserved in that region. So they use donations and work with stakeholders locally to buy up land or get easements on land, conservation management throughout this region. And it was really interesting talking to him and standing there in Podocarpus National Park. We were looking over to the other side of the mountains there, one of the most biodiverse regions in the world, by the way, in southern Ecuador. And I asked him, so he says, yeah, we got this land conserved over there. And so I asked him, so do you know what you're conserving in it? Do you know what's in there? And he said, well, we know about that species, and we know about that species. And but that was it. And we sort of started to talk more. And it's obvious, actually, that they have, with the limited capacity they have, it's not their key thing to necessarily find out what lives in each of these places. And equally, as they make decisions on which places to go and conserve or do triage, well, should we go for this one and drop that one, or et cetera, they actually go with very, very limited information. And to a degree, that's totally fair enough, because you want to be opportunistic and move in before bad stuff happens. But at the same time, it was clear, and he made clear, that if they only had more detailed information, they would be absolutely excited to use that in their decision-making. They don't have the science capacity by themselves, but they would really be delighted to use it if they could. So that was kind of really enlightening. And equally, there are other initiatives going on in South America. We just had Half Earth Day in New York a few weeks ago, and I was sitting with somebody in a panel from Gaia Amazonas here. There's all about engaging indigenous communities in the challenge of a more sustainable development in this region. And here are all these indigenous areas, and again here the case where they would love to know actually what biodiversity is in them, which are the sort of priority regions from a global perspective in terms of preventing species extinctions and ensuring the maximum retention of biodiversity into the future. So this is where Map of Life is trying to offer a service, offer information. This is a project we founded here like 5 years ago or so now, with initially NSF funding, and then we've been able to fundraise through another number of other places, and most recently through an engagement from the Half Earth project and donors associated with that. And that's really trying to address these pieces that we just talked about. The question that Renzo had, well, yes, if you could give us more information here, we'd be delighted to use it. And we are at a day and age now where new methods, new data, new technologies are able to put an immense amount of information into this equation. So Map of Life is trying to do this essentially. It's trying to bring together these disparate, link up these disparate sources of information that go way beyond museum specimen. That's an outstanding source, and we have some of that information just in the other direction over there in the Peabody Museum and many other museums around the world. But more and more, that information gets overtaken in terms of magnitude and by citizen science data, by other sensor data, camera trapping data, movement data, and I'll say a few more words about these things. And we have already existing initiatives that are bringing some of this data together for certain communities. There's a lot of information in the literature. So we've been aiming to link this up and attempt then a first order, at least visual integration. So we appreciate how the information gaps, the data gaps, and then link this up with environmental data to produce spatial and spatio-temporal predictions of actual species occurrences in space that could then be fed out again to individual users or more programmatically to some of these larger scale policy processes and conservation NGOs. So you can look around the Map of Life website and look at the range of data sources, and it's growing every day. And that's not just your point data, it's all these other data types as well, range map data in particular, expert maps that you may know from your field guides, et cetera, et cetera. So we can then put this all together and aggregate it up to calculate, for example, range verity. It's just if it's a map of endemism, how range restricted species are, right? That would sort of prioritize places that if you did conservation action there, that would be a key place to prevent future extinctions, as certain species would just be restricted to that location. But it's pretty pixelated. You see, it's pretty coarse grain. And that's because the information that we've been using so far is very coarse grain. It's these blob maps that you see in your field guides that you can't just use at a finer spatial resolution. And you see that, however, even with that information already, there is a lot of data, a lot of gaps, places that seem to be of high priority for terrestrial vertebrates and a few plant groups, yet are outside established conservation areas, or in this case, community-based protected areas. Now, we can go in with a bit more detail here. This is work we're doing with the Field Museum, with MacArthur funding we received, and I was just up there a few weeks ago, where we were reviewing and discussing the next steps around this dashboard, where we have used some of the data without models yet, just some of the existing data to do reporting, simple reporting, even with just the basic data we have right now. So you can look at these 3 countries, that's our focus right now, and go in and, well, learn about the number of species according to these expert maps that I talked about earlier are occurring in Peru, and that's quite a lot of species. It's one of the most biodiversity countries in the world. You can then learn about which ones are nationally threatened. So we are getting to a sort of an information tool here that may begin to have some policy or local decision-making uses. But this is where it gets more interesting. We're then breaking it down by counties or by watersheds. And obviously, we coded this website up also in Spanish, so it's multilingual and can be used locally. And we're working with the national park agencies and individual park managers to make it useful for them. We have then pulled in information for all of the parks. And this is not yet detailed, accurate information. It's simply like given the general blob maps that we have, these expert maps, where experts think a set of species would be occurring. And we have that information for thousands and thousands of species. What do you think is occurring in this particular park? So it's kind of the maximum number of species. It's the expected species. So and quite a large number of birds, mammals, reptiles, etc., etc. And by the way, that same information, the same sort of idea is in the Map of Life app. If you want to play with it, you can find it on Android and the App Store for iPhones. You can find out what's around me or what's in this place, and you get this sort of initial species list, and then you can drill in and learn about those species. You see photos, you can do species discovery. It's kind of this idea of a flipped field guide in a way with the data and the geofencing that can happen through this. We can tell you what you're going to be able to see potentially, rather you having to flip through a book. But more importantly, what we've done here is we've contrasted this now with which species have actually any sort of local observation against them for that park. This is the publicly shared data. What I showed you earlier is just an expectation, right? That's the expectation. It's not even model-based yet. It's just one data type. It's an overestimate. This is 12 records of bird species or have been recorded and shared from that location. And now you see the dilemma of national park managers in that region. They may actually be managing a park, and perhaps themselves they may know a little more than this. But generally, it's not in an organized. It's not put together in an organized way. It's not usable. It's highly biased to the species that are easily observed or that museums may have readily picked up near a roadside. So this is kind of the challenge that we're dealing with in spatial conservation decision making. And here, all we did in this dashboard tool is kind of put these two pieces together. And so you can now already imagine the next step. We haven't done that yet, but you could begin to color, for example, have a map of each of these parks and show which ones have the greatest gaps, which ones are of highest priority for additional data collection. Right? And it helps to have this expectation, because in some places you actually expect, well, we expect about 100 amphibian species to occur here, and that's a different expectation or benchmark to work towards than in terms of your data collection than another place where you may only expect 5 or 10 species. Yeah? So this can be very powerful in guiding future data collection activities and appreciating uncertainty or lack of actual data as you make decisions about certain places, and do we extend that park or do we put a park next to it or somewhere else or any sort of other conservation decision. Now we've formalized this in an indicator, the Species Status Information Index. So this is a formal core indicator of FES. It's one of the 30 core indicator in the global assessment that's going on right now, and it's also part of the CBD process and discussions. So ultimately something that nations may have to report against on the target 19, which is about the growth of knowledge, how our nations improving their understanding of biodiversity. And what this is doing here, it's looking at, so instead of a park or a county, we're looking at equal-sized grid cells, and what we're mapping out here is simply places where nearly 100% of expected species have been recorded in situ, or down here, places in blue where close to 0% have any sort of raw data against them. And you can then see the growth in this ratio that I just mentioned, so observed divided by expected. And in some nations, such as Colombia, where there's a lot of really good activity going on, there has been a strong increase in their countrywide biodiversity data coverage. And you can compare nations and you can compare regions. And in all parts of the world, thankfully, we have been improving on this indicator. But in some parts, we're doing much better than in other parts. So South America particularly lagging behind compared to other places. So back to the idea of Map of Life, or of others, of course, the larger idea out there that we're trying to operationalize in Map of Life, of integrating, combining different data types. And here you see one kind of typology, if you will, of different data types. You have these expert range maps that I mentioned. They are very coarse spatial grain. You can't expect to see a species only because you're inside that blob, right? And however, we have pretty good availability of that information for quite a large number of groups. And then you have these other types of data, area inventories, focal point records. These would be your typical citizen science records, etc., etc. So, and there are more intricate data types down here that can also help us fill in spatial biodiversity knowledge, right? You may not have much data for this species, but you may have a lot of data for its close relative, its sister species. And we expect those niches to be quite similar. Actually, you can model that on the tree. That's another piece of research that we've been driving forward. So if you can characterize this species well, that actually may tell you a lot about this other species that lacks in situ data, but at least you know the phylogenetic association. So these are all pieces we can bring into this equation, which is really powerful. And then we need to, for this integration, there's a lot of brunt and sometimes not most exciting work that's necessary. And that one key piece to this is actually standards. It's standards, standards, standards. If you want to be able to combine different data types, you got to have fields that you can link up, that the meaning is standardized of. And one key work that we've been advancing is that of characterizing inventories. So any of these data types here, checklists, atlas surveys, time series surveys, citizen science checklists, like characterizing them in a standardized way so they can all be integrated, something we called the Humboldt core, with the same the same standard, sort of standards for point data being named after Darwin, the Darwin core. So this is pretty technical stuff, but it's important. The other pieces we are really excited about is this new type of data coming about, animal movement data, for example, and in particular, initiatives such as Icarus that essentially has put a huge telemetry or GPS tracking receiver up on the space station that now allows us to scale the idea of animal tracking to nearly the whole globe. So when initially, yes, increasingly we were able to work with smaller tags, but initially you had to get close to read out that tag, read out these GPS data that may be on that tag that you put on a bird. Here work by graduate student Catherine Mertes in East Africa. Instead of having to get close to that bird, you can now just read out the data via the space station. And the antenna got installed this summer. It's really exciting. It's kind of ecology, finally gone to a big machine. And we're all really, really happy and excited about that. And we're really looking forward to the new types of and new amounts of data that will become available through the system. This is not new. This is existing data. Already shared existing data in MoveBank, which is a place that brings together movement data from all sorts of providers, that shows how animals, mostly birds, connect the planet. And it fills in a picture that we just haven't seen before. And this is just with existing data, with old technologies, and we expect for this globe to become all read and filled in, and for us to be able to start to collect a baseline about how animals, mobile animals, currently use the world. That can be of real fundamental importance going forward, that we can then look back at 10 years down the road, 20 years down the road. How have animals' use of the landscapes changed? How has it stayed the same? We can even imagine real-time alerts as animals from 1 week to the next or 1 year to the next are using this forest in a different way because perhaps it's not a forest anymore, right? And we can suddenly work with remote sensing data in a much more biologically interpretable way because we have these things moving around telling us about what habitat actually means for them. So instead of having to rely on human-driven land cover classification, we can begin to imagine something where animals, as they're using the space or changing their use of space, tell us about what this remote sensing signal actually means for them. And we've begun to do some of this work with storks, and here an annotation of a greenness index for a single individual, actually no, multiple individuals as they moved from Europe to Africa. And here, just to convey to you that much more than the GPS record, we're actually increasingly able to use animals as well as also as a sensor. There are already temperature, humidity sensors, other sensors on these tags. And this is obviously not something that's scalable quite yet, but in the future, you can imagine that animals will be able to provide us with more and more information about not just themselves and their behavior, but also about the environment around them and how it's been changing. And this is the sort of stuff that we're really excited about as part of the Max Planck Yale Center. that we launched earlier this spring, and actually we had a symposium in this lovely room, we're working closely with the lead and founder of Icarus here on the left, Martin Wikelski. He's the co-director of that institute. He's co-director of the center and is leading an institute, the Max Planck Institute in Germany, and we're really excited about the future here. We've begun to recruit postdocs. There's one of them sitting here in the audience, actually, that are going to solve all these exciting questions. Here, a look back at Humboldt there, and I'm pointing out these instruments because I think Humboldt, if he was to come back today, I think he wouldn't be much impressed by where we are in our understanding of biodiversity on this planet. I think with so much time gone, I don't think he'd be impressed. But he would be excited about the new toys compared to his era. And I'm certain he would jump on them, and he would be out GPS tracking and using remote sensing data, and that's, I think, what we should be using, right, and doing. And let's talk remote sensing. So here just the NASA, a selection of NASA satellites, Earth-observing satellites that are relevant for us. Actually, I think there are some others in there too, but these are all key pieces of information. And so far, actually, ecologists haven't engaged so much in what is it about this huge flow of data that's coming from these satellites that is most informative about biodiversity. So with NASA funding, we developed some new types of layers, and I was surprised nobody had done this before, but cloud information, this cloud flag that's usually seen as a nuisance, is extremely powerful to tell you about, variation in cloud cover around the world in great detail, and in its seasonality, in its among-year variation, and it's able to tell us in an observation-based way, not just interpolated or statistically modeled, in an data-observed way about variation in cloud cover and the associated variation in vegetation that otherwise you just wouldn't be picking up from this side of the mountain to that side of the mountain. Look at how sharp this ridge is up here, for example. Any of you that have been to Monteverde or Central America would appreciate this is a razor sharp there, and indeed, when you're up on that ridge, it is razor sharp. You have the Atlantic influence on one side and the Pacific influence on the other side. So remote sensing data is able to help us here, and, for example, allows us now to map out cloud forests in a much, much greater detail than previously was possible, and allows us to monitor them going forward. So here, a high-resolution map of cloud forests worldwide that we can now use in our modeling and in our change assessment. Now, so this is sort of straightforward, if you will, environmental data that we're able to pull in, but we now want to take this to a more advanced level with models that we really link to in situ biodiversity data, and that's where another project is coming in that we doing in support from NASA, and don't get lost in the details here, just to say that the combination of in situ observed biodiversity data and remote sensing data is actually not super trivial. They show up in different spatial and temporal resolutions with different spatial and temporal uncertainties, and it's quite a lot of data. And you may not just want an average, but you may want the information on the day of observation, or from the precipitation the 100 days prior, or you may want to statistically grab that in the way that's most informative. And so we're building infrastructure that would allow doing this in a general way with all data that gets collected and shared to Map of Life, as well as any data that users may want to bring into the picture and then can feed out into all sorts of applications. And what that allows us to do is to then not just show a geographic map of a species. This is the Van der Deckens hornbill that Catherine did her PhD thesis on, and all sorts of citizen science and museum data you see there, these points around the safari route. You see that's where a lot of data people are sharing their data in Kenya, Tanzania borderlands, much less data in northern Somalia. But the blob map, this green map, tells us about it. Well, the expert says the species does occur up there, you just don't have a lot of data for it. Now, what we're able to do now is characterize both these data types, and here is just shown the points, not just in geographic space, but also in multivariate environment space. So we're able to look at the distribution of that species, in this case, precipitation and temperature space, according to this data. And we're now able to do that for every species in Map of Life, tens and tens of thousands of species. And you're able to then, look at this niche, and you're also going to be able to monitor it going forward. But more importantly, we're able to include this information in environmental niche models or species distribution models, where we then take this multivariate niche to separate presence from absence, and then put that back on the map. And we're able to increasingly do that, not just with one data type, but thanks to work by Cory Mero and Adam Wilson in our lab, by integrating different data types. So here in black outline, you see this blob map for the solitary tinamou. So this is where experts say, well, I believe roughly the species occurs here, but this is lacking detail, right? Now we are getting the detail from the point data, and in this model, we're now combining the best of both pieces of information. So we take the high-resolution environmental associations from the point data, and we take the broad distribution limits from the expert map, and we're essentially constraining our prediction in this way so that we are getting to a much more reliable map where we're excluding points within the expert map that are definitely environmentally unsuitable and are able to constrain predictions based on what the experts are saying. In another step, we brought in expert elevation limits as well, as exercised here, exercised through for a hummingbird. Here the different data types, a national park checklist, point data, expert maps. It's actually a pretty well-sampled species throughout its range. Hummingbirds get observed by bird watchers heavily. Through our niche model, here done by Diego, a graduate student in EB, we were able to do an integration and now put this to much higher spatial resolution. And as you zoom in, you see how all of these data types on their own would have missed or over-predicted the distribution here, or altogether missed the distribution there. And suddenly, we're able to provide a much more detailed representation of where the species actually occurs at a kilometer resolution. And if you're doing decision-making here about where to place a reserve, or you're wondering, does this occur here or does it not occur here, this is the sort of detailed information that you require. So we can begin to take this map that I showed you earlier for a select group of species to much higher spatial resolution. So for hummingbirds, we've done this now, this detailed map for all species. And you can go in again and now look at this rarity map. So where are the range-restricted hummingbirds in much greater detail? And you can identify specific ridges or parts along this mountain range that may represent conservation gaps for hummingbirds as a whole. And going forward, we are repeating this, we're expanding this to all bird species, and want to go beyond that to other species groups. And just to show you what that looks like for more species groups and in practice, here for just South Africa, so this is not addressing the whole global range of these species, it's just addressing the South Africa piece. For birds, mammals, and proteas, highly endangered and unique group of plant species, South Africa. I'm showing here on the left species richness, on the right their patterns of endemism. And we need to appreciate here that had we only used one group to identify hotspots of endemism, we'd probably missed quite a bit, right? And a key challenge now going forward is to not just do this for one group, not just for hummingbirds, not just for proteus, but really become as inclusive as possible. And that's why we've partnered up with the Half-Earth project out of the E.O. Wilson Biodiversity Foundation. So some of you may know Ed Wilson's book, The Half-Earth, where he's advocating for putting more of the planet making more of the planet a priority for conservation, and in particular, think about more species, include more and more species. Think of the species as a conservation unit. So this becomes clear as you look at these maps, and as we zoom in, we start to appreciate something else, that we're now actually looking at biodiversity patterns that are detailed, where we can bring it together with potential drivers of change or pressures. We can think about, for example, the parts of South Africa that are already human encroached, that are agricultural or urban. So these are some of the wine growing regions here. This is Cape Town. And we can bring the existing conservation management areas into the picture and begin to think about, okay, where are the conservation gaps here? Where are opportunities? We can begin to feed this to local stakeholders, to NGOs that are making decisions about where they should be active. We can tell them about, hey, this is what we think occurs over here. In many cases, they will already know, especially in South Africa. But in quite a few cases, for quite a few species groups, they may actually not know. And they may not yet appreciate or be able to argue with their with other stakeholders the global significance of some of the places that they're considering or that they're concerned about. And we're able to bring this in. And here actually some really successful private reserves for tourists, for proteo watching, for example, that have been put into place here, and they're thinking about expanding. So this is all very real. We can feed this information into local decision making if we prepare it right and then offer it up in the right way. So the larger ambition or vision here is that we are hopefully able to get an increasingly rigorous and spatially detailed and taxonomically inclusive evidence base into conservation decision making, are able to provide indicators about how well we're doing achieving certain conservation targets around biodiversity rather than around just areas. But we can also do run smart algorithms about optimum reserve design, about spatial conservation prioritization over this data, and offer this up in a way that allows a back and forth and an iterative updating as new data comes in, and therefore offer up scientifically rigorous yet really powerful decision support for some of the actions and management happening at the local level. And I think looking at this here, we are actually, if we are able to fill in this map and this stack of spatiotemporal biodiversity predictions, we are actually able to measure how well we are doing. So how many gaps remain, how many species are being left out of the current conservation management areas. And I would argue, and I'm trying to advocate for a target that's not just any more area-based, but that is thinking about the species, about the conservation coverage. So instead of a certain percent area, that doesn't make sense because you're putting the UK and Brazil on the same footing. If we really want to be representative about biodiversity, we should put biodiversity into the equation. And I would argue it's conservation coverage commensurate with nations' stewardship that would ultimately be the most powerful and ultimately possible at this point. So with this, I want to just mention one more time the Half-Earth project that we've been engaging with, and thanks to support from Paul Simon, not just in terms of singing to us, but in terms of actually financially supporting this research and others. We are now going forward in trying to do this work, trying to provide these high spatial predictions for conservation decision making with GeoBon, and I'm really delighted that Melody Megochi is here. She's the co-lead and co-conspirator of the Species Populations Working Group in GeoBon. We're also trying to think about this temporal axis here. So biodiversity distributions are obviously not static, they're dynamic, and this is our conceptualization. We don't want to just fill in a map, ultimately we want to fill in a cube, so the space-time distributions of species. And with that in hand, we have something really powerful in terms of indicators over time that we can feed, that we can support through some of the basic science happening down here. So with these pieces in place, you can look at both spatial distributions and how they're changing, or abundances, or whatever it may be, and you can look at trends over time. And then here's just one example in collaboration with National Geographic and others. We've been advancing the species habitat index in Map of Life, where in this case we are working at the same species as I showed earlier. We're starting with this blob map, but I'm not going to show you this model that I showed earlier, this inductive model driven by remote sensing in terms of a statistically driven association. Here we're just putting in, well, we know this species is a lowland species and it's dependent on forest. Over the last 15 years, only the red and blue pixels have ever been suitable. because those are the only pixels in lowland and forest, and we are able to map that now in high resolution through remote sensing forest, land cover 30 meters. And in fact, these red pixels have all been lost in the last 15 years. So the species, we are able to estimate its range through some of the other data we have, its range size in detail, and we're able to show how much range it's actually been losing in that region. And we're able to do that for thousands and thousands of species, and we're able to aggregate that up to the country level and use a stewardship weighting in that context that essentially says, well, if you have species that are only occurring in your country, it's actually particularly bad if they're losing range. If they're species that you only have a tiny bit of their global range, then you have less stewardship, and we're not going to weight that quite so much. And this is essentially just looking at decreases. Obviously, there have also been increases, but looking at just decreases, this is how nations across the world kind of map out, with some really disturbing cases down here where a lot of species have lost quite a bit of range over the past 15 years. In related research, we're using projections, and now taking this forward, we're looking at, again, this distribution in detail here, and this is already in Map of Life. We just haven't switched it public yet, as is the other visualization that I showed you. So this is all going to be accessible for every species. You can interact with it. It's transparent. It's not black box. You can look at the species by species case, and here we're looking into the future. We're taking the latest land cover projections that are part of the range of socioeconomic scenarios that is part of the IPES and IPCC associated modeling of the future of our planet, both in terms of climatic conditions and land use and land cover conditions. And for some of these species, we are projecting dramatic range losses. So we can use this in both directions and then make projections about which species may be, and which regions may be particularly threatened in terms of land use change translating into high biodiversity extinction risk. So with that, just the final slide, bringing it back again. So I hope I was able to show you how some of these new data methods, technologies are allowing us to provide a lot of new pieces to conservation decision-making and policy. But I want to make the point here in ending that they're also providing a lot of exciting pieces back to our basic research, and that they really can inform each other, cross-inform each other. We're able to go across these scales now with some of these global scale yet super detailed pieces of information and data. And with that, I'd like to thank the funders, and then particularly the members of our lab group of Map of Life, and then several of them are here in the audience. Big thanks to them for making this all possible, and then thank you.
So on my front yard, I have monarch butterflies fly by. So I'm wondering how I interpret this with large migratory species, because by making a decision just to plant another butterfly bush or not use pesticides, I'm dependent on all these other activities going on over the whole range. And I noticed a concomitant, you didn't really talk about the marine sphere, so I'm wondering if they're also very migratory, the fish, so I'm wondering how you deal with migration.
Yeah, no, the temporal dynamics are vast in some species groups, birds, butterflies, marine. And that's one of the key pieces we're hoping to address actually with this new data type, this global GPS tracking. Now, partner Martin Mikelski actually just managed to track some moths for 3 days on his plane. First case I know of any sort of distance of butterfly or moth tracking, in this case Lepidopteran tracking, and not quite ready yet for prime time. And it didn't use Icarus; it used his plane just following around, listening to the pings. So I would say one answer is the stork example I showed you, for example. That's work by Ben Carlson, a graduate student also in EEB, that's trying to map the migratory distributions, the seasonal distributions of storks in great detail at the level of individual and scaling up to the level of the population and the species as a whole. So that's when we have GPS tracking data, really great new opportunities to tackle it that way. With butterflies, it's a bit of a more difficult issue. We're lacking data. However, citizen science data is tremendous for some of these species groups. And actually in monarchs, I wouldn't be the one standing here telling you about monarch space-time dynamics. There's actually a couple of NSF-funded projects that are doing really outstanding work now, integrating remote sensing and citizen science data to model the space-time distribution in detail. So they're able to project when they may be moving through, and they may be able to tell you in great detail which types of habitats along or places along the migratory route would most benefit from like planting milkweed or other things to support them. So I think it's one of the larger challenges, these highly temporally dynamic, spatially dynamic species, but it's equally going to be informed increasingly strongly by some of these new data.
The technology is really impressive, and the fact that you're able to identify all these areas. But to put this into policy depends upon everyone being middle class or higher, and that's not really true in many of these areas. And you could adopt the annihilation of the poor philosophy, but that denies the fact that poor people around these conservation areas that live off the land are going to move in on them. And so I think there's a real problem. It's more of a sociological problem, but it's something that limits the use of this kind of data.
Absolutely, but I think where we can be supportive, so this is in the realm of local government, local NGOs. So we are not an advocacy group, our lab, or we are not a conservation group. We're trying to provide best possible evidence supporting conservation organizations. I would say that this increasing ability for real-time monitoring or alerts could be really powerful in this regard. So you can actually help regionally active NGOs or folks with a system that would alert, hey, in this high priority area, this whatever deforestation just happened. And it's not just any deforestation, it's like in this very place where we expect these really rare species to be. Or with GPS tracking data, this place just has been avoided. It was always been used, now it's suddenly avoided by our sensors, our sentinels out there. And I think that could be a really powerful way to bring science and technology into reacting most effectively to the sort of issues that you brought up. And by the way, I should say, there's also an amazing educational opportunity. I mean, we've been amazed how much people, and not just our app, other biodiversity apps out there, with this information, we're able to engage people in a new way with biodiversity. It makes it easy for them to connect and identify biodiversity. So I think that's a general appeal that can help out also around the concerns that you raised.
So I see a couple of hands, but we're unfortunately out of time. The good news is Walter is on campus. He's in that not-so-beautiful outside building, but clearly he's got a really exciting lab, and I think he's really open to getting our FES students involved. So please join me in thanking Walter for a great talk.
Did it thematically fitting enough?
A lot of our faculty here. It's good to see our dean here. And,
Oh yeah.
I think that was perfect.
Right, thanks. Well, thanks for having me. Yeah.
No, that was terrific. And I think it's also good just to see our students, our affiliated faculty here.
I'm so happy to meet you. My name's Melanie Quigley. I'm.
Oh yeah, you were helping in the setup. Yeah. How you doing?
My husband's also Brendan Smith. He was doing solar on your house. He did solar for Karen's house.
Yeah.
I'm so happy that you're working with Helen, which is here as a guest speaker.
Helen.
Did you say her name?
No.
Thank you.
Working on wildlife.
I'm gonna have to take him.
No.
Okay,
no.
but I do
But
want to
yeah.
say that we're sending a bunch
But he
of
is
folks
just over
coming
there.
forward.
I
It sounds
take
like you're
him.
going. Do you have any other folks coming from your team? You're going.
That's me.
So
Yeah.
I'm going to add you on to some.
Oh, cool. You're supporting some students again to
Yeah,
go?
so I'm going to go, and we have a booth for Yale. We're
Oh,
gonna have
sweet.
a